this morning, I would like to take you through just a research compliance process. I was told to do manuals and SOPs, and as I told you yesterday, I'm sure we have the template of a manual, we have a template of an SOP, and all of us, because from what I've heard from all of you, you've been in research. So all of you have SOPs, true or false? Don't we all have, just show me, don't we all use SOPs? Otherwise, how are you doing your research? There must be a protocol. Some of us call them SOPs, some of us call them protocols, but they're just all the same thing. It is just a step-by-step way of doing your project. So all of us have some SOPs. There are many suggestions on how to write them. I keep on saying there is none that is superior to the other. The key thing is to have a documented procedure. And a documented procedure that you can validate that works. And then you write it down. So that if I come to use it, it is reproducible. That is what it means to have an SOP. When it comes to a manual, a guideline, the word is guideline. A manual or guideline gives guidance. It doesn't tell you what to do. I hope I've been able to tell you the difference between an SOP and a manual, because that is all I wanted to do. Then I go to the process of research compliance. So a manual is a guidance document like the WHO Biosafety Manual, guidance document. I was looking at the Uganda National Council of Science and Technology, your guidance document on IBC. That's a guidance document. It's not cast in stone. When it goes to the institution, the institutions use it as a guidance document and they write their own manual as long as they comply with the things you've written in your guidance document. So this morning, maybe I'll just pick up quite a number of these things I could have said. I might have talked about them yesterday, but I just want to take us through a research compliance process. And for you to go through what you call research compliance, and just allow me this morning, I don't like the approach of picking one thing, recombinant DNA, and just talking about it. When you're doing research, there are many elements of compliance for your research. And we've been talking about quite a number of them. I think as we've talked in this room yesterday, a lot of you have talked about ethics. And even in the discussion on gene drives, you heard about ethics. So ethics is one of the elements, isn't it? If you work with animals, ICO could be one of the elements. If you work with biological hazardous material, IBC will be one of your elements. If you are introducing material into the environment, EIA, impact assessment, will be one of the things that you have to do. So there are many, many things. If you introduce a new breed of animal and say it's superior, you need to do an impact assessment and show how it is more superior than the others that you are saying it is more superior or is producing more milk or whatever you are claiming your new breed is doing. So there are many aspects of compliance. When you are moving materials, I think Labi went through a very big exercise that you're saying they thought they had done everything until they needed to move material from a lab that meets compliance in public health to a lab that is under research and development. Then they realized this research and development lab does not have what they have. So how do we move to a lab that we are not sure they can handle the materials as good as we can handle the material? And you get into a lot of conflict. So all those things is good for us to understand. They are all part of compliance in research. And there is none that is more of higher value than another. You just must meet all of them for you to be doing good science. So one of the things that I've seen works a lot is to start by what we call a policy. So when you talk of a policy is a statement that your institution makes. Not us as a group of scientists. We may be the ones that influence our institutions to write these policies. But you must make sure the institution that you work in, does it have a policy on research? And does that policy of research include research compliance? Does it talk about doing safe science, good science? You know, there are many words that are used in those policies, but are they used? Because that is where you are going to anchor your compliance issues. So you have to make sure that your organization has a well-defined compliance policy. And I don't know, do we have them in our organization? This morning I'd like us to talk. I want you to talk back to me, please. Just talk back. Do you have it? Do you have it? Ghana? This is Tanzania. So do you have a policy that is for research? Does it include compliance? You have mics on your tables, by the way, so you can talk and everybody can hear.
Yes, we have what we call an administration manual, and under that there is all information regarding operations in terms of admin and the research side of things. And there is some, I would say, documentation regarding research there, yes. I don't think it's as comprehensive, maybe perhaps as what you've explained yesterday, but we do have a research policy in our institution.
So let me challenge all of you. Go and read the document that outlines research in your institution. If you are a regulator, it may be what creates you as a regulatory. It has something to do with compliance. You know, sometimes I keep on saying we think regulators don't meet compliance. Don't you meet compliance as regulators? You are here. Do you meet compliance as regulators? Mushiri, you're always hitting me on compliance. Today is my day with the microphone. Do you meet compliance as regulators?
Maybe you can repeat the question, Effie.
I'm asking, as regulatory organizations, do you meet compliance?
Yeah, of course, someone said yesterday, compliance is progressive. Yes, we attempt to comply with, like for example, for us, we try to comply with the Act and also the regulations. Yes, but it's progressive.
Yeah, so you have a document, you have the Act of Parliament that does not even give you a chance unless it's repealed, isn't it? So that's your compliance document, yeah?
So in regulation, the word is not meeting compliance.
It's what?
The word is being guided by a complying instrument.
I'm telling you, I'm talking to scientists, specific, yeah, very specific with your words. But all you are doing is there's a document that guides you. And it's not a guidance document. You have to go by that document. Because when we talk about an Act of Parliament, like the one he has referred to, it has given prescriptions of what should be done for GMO work in Kenya. And he has to go with that. I've seen the same with the countries that I've read. If you have a proclamation, some of you call them proclamation. The other word that came out yesterday was what? Degrees. They are very clear. The degree says we shall have this and this. Before your project is approved, it has to go through this and this. Some of them, I heard the Uganda gentleman tell us that you needed even 75% and above of the voting. Those are things that you cannot just wish away. You either do them or they will not give you permission. So that's what I'm saying. I just want us to all understand that we all meet compliance. Whether we are regulators or whether we are in research, there is a way we meet compliance. And that is guided by some of our policies. So if you go to our policies, they will talk like, and in regulations, it will be what created you, what is your purpose as a regulator. It will be guiding you to which law are you supposed to implement. So at our institution, at ILRI, what we do is we've realized that there are a lot of regulatory requirements. As I told you yesterday, it's like a maze. You know, you've just been thrown into something, and you need to find out. And from what you people have been talking from yesterday, I used to think Kenya is different. I've worked in most of the African countries, West Africa, Eastern Africa, Southern Africa. We are all the same. Regulators don't talk to one another in our countries. Isn't that not true? If you go to NBA, don't expect that they will tell you every regulation to do in Kenya. They will tell you about how to comply to working with recombinant DNA. Then when you move to the environmental regulator, they will tell you about access and benefit sharing. I'm just quoting the ones that affect us, ABS. Then when you move, like for us who work with animals, you move to our Department of Veterinary Services, they only talk about how to work with animals and move animal material. When you go to the plant regulator, they only talk about plant. You go to wildlife, they talk about wildlife. You go to public health, they only talk about working with human beings. So that is how our regulatory framework is. So as an institution, you have to have a policy that helps you come up with ways in which you are able to meet regulatory requirements and best practice in the conduct of your research. And yet regulation is big. And that's why you must have a way in which you map your regulatory requirements. So I know we are in a workshop where we are working with recombinant DNA, but that is not the only science regulatory requirement. There are quite a number. So once you have your own policy and you have tagged that policy at regulatory requirements in the country where you work and international best practice, then you write your own internal regulatory requirement. We always say internal must be a little bit more stringent than country level, so that you are always at a safe operations. So at the minimum, all activities in your institution must meet your internal policy. And I think most of you do that. Whatever is the internal policy in your institution, whether it's stringent or not stringent, because if your institution says, those of you who are talking about ethics, it's because your institution is strong on ethics. True or false? They insist that before you do, and for the people who do health research, you cannot even start. Am I right, the health people? So that is why you have strong ethics, because the regulatory framework in health is ethics. It's research ethics. So without research ethics, even nobody will look and approve your project in health. So they are more strong on health. If you work with animals, you may be talking too much on ICOOC, animal care and use, because you cannot publish an animal experiment without an ICOOC approval. Now, for those of you who work in labs with biologicals, then your key issue is you want to work safely with biologicals in the lab. So you might be much stronger on IBC. And that's why you are discussing here. And that's why you are asking, what about emerging technologies? How can we do them safely within the labs and not be of any issue? I think the key thing you are discussing here on GeneDrive, if you think about the key things you are discussing, they're environmental. True or false? You are discussing environmental. I didn't hear any risk to a person. I kept on hearing more of what I would call environmental risk. So those are the things you have to think about. In which scope are you in, and make sure that your policy covers those things. One of the things that I have seen a lot with the scientists, and I saw this a lot, I see it even with our own scientists at ILRI. If our scientists came to this meeting and met Mushiri here and discussed with him their project, and Oshiri says, that is okay. That one looks like it's okay. It meets the requirements. It does. Mushiri might even tell him apply, but what did our scientist hear? He will come to ILRI and tell me, I talked to the deputy director and he said, it is okay. And if he talks to when Dr. Tonoi was, Prof. Ogoi was, they just chatted with Prof. Ogoi. And he said this work, in fact, that is what they are waiting for in this country. You know, we've been talking GeneDrives and saying it should, you know, what can we do about it? It's a good solution. Then it looks like the regulators were all sitting here, and you go back home and you say, the regulator was there, they seemed like they were happy with this thing. And people just go to the lab and you begin, isn't it? So that is why you must have your internal policy, which now leads you to the regulatory agencies. Are you getting it right? So every regulation must start with your internal policy. How do you speak to your people who do research about research in your institution? So when you come at ILRI, and this is in most institutions, your approval for research starts internally. I hope I'm right about that. You don't just pick a project wherever you are and you start. Whether it's a university research institution, there is an approval process for your research. I'm right about that. Somebody says, yes, go ahead. So there is an internal, and then there is approval that goes at the next level, which is the country where you're doing work. If you see my presentation, I say host country, because we do research across countries. So you might find our approvals. I'm looking for GMO approval, contained use in Kenya, field one in Uganda. So you're looking for different approvals in different countries. So once you know this is where I'll be doing my work, if you're doing in your own country, then you look for approval, the host country authorization of research. And that is mainly through issuance. The first step is you need a research permit. And this is where the process is very complex in countries. Who is responsible for research in most countries in Africa? Which institution is usually responsible for research? Any research, just research. When you start talking research, who is responsible? Higher education, which other one? Commission of Science and Technology in most of our African countries. So when you go to research, it's either higher education, science and technology, whatever name it gives it. But it puts you on the same people who regulate the higher learning institution. That is where mostly research is anchored. So those are the people who will give you a permit to do research. And that is the starting point. You need a permit to do research. Now, I will give an example of Kenya because I've applied many. I might give a bit of Uganda, Tanzania, because we've done permits and a bit of Ethiopia. So if you're looking for permit to do research, one of the things here in Kenya you'll be asked for, and it's the same thing you're asked in Uganda and Tanzania. And look at it, countries actually speak to one another, influence one another. You start seeing them behaving in a similar manner. The first thing they ask you, if you want to do research, they ask you for an ethics permit. The regulators, Uganda, Tanzania, isn't that what you ask for? You want ethics approval. Has the science been reviewed? Is it good science? Who reviewed it? And most countries are now saying the review committee must be registered in their country. So you have your internal committee on ethics. You have reviewed internally, you've approved, but now your internal review now needs to go to the next level in the country where you're doing your work and get approval in that particular country. Now, if you are ILRI and that project is in 6 countries, it means you're going to get 6 country approvals. And each of them might go through an registered committee in that country. So that is the process, and that is just for you to get permission to carry out research. You haven't even gone to the next level of saying, now what else? Your research may affect other things, but you are just getting permission to do research. Then on top of that, you may need additional authorization. If you are getting money from anyone, most donors give you, and that's why some of you were talking yesterday of NHIF. Why are you told to comply to it? You have American money, true or false? That's why if you have Australian money, they'll tell you to hit. If you have any EU country, they'll tell you to do the EU regulations. So those are the 3 key areas that you find yourself that you must know what are the regulations so that you are in compliance with all of them. So internally, what are the pillars of compliance? Because there must be some pillars that you cannot, if we have this in place, and you don't have to call them that name. You just need to have them in your organization. You must have a pillar on research ethics. all organizations, you must have a pillar on research ethics, animal care and use if you work with animals. And then you must have a pillar on biosafety, biosecurity, and you must have a pillar on how you do your research permits and licenses. Because you get all those, but you still need your permits and licenses to move material, to do research. Many of them arise. So if you have those pillars and you have them right, and everybody in your institution knows which is the ethics pillar, which is the animal care and use pillar, which is the biosecurity pillar, and which is the research permits and licensing pillar, then you can tick your boxes and say, we are almost at meeting compliance in doing our research. Because once your people know those places are there, they are clear on how do I get information about them, then now you can do your research and say you are meeting compliance. So what does the institutional research ethics really do in an institution? They actually will do the scientific review for all projects. And I think when I talked yesterday, that's what I said. All projects go through what we call scientific review. And we all agreed, whichever way your project goes through a scientific review, whether that scientific review is done by one person called your supervisor. You tell him, this is what I've found I want to do. I've been funded, whether you've been funded by your country or you've been funded by an international donor. You go to your supervisor, you tell your supervisor, I want to work on gene drive. Your supervisor reads it and decides, it looks like good. That is a scientific review. It may not be meeting the threshold, but that is what your institution is doing. So sometimes people say we don't do it. You do it. Some of us are not doing it well, but there is a level of doing it because somebody must approve what you want to do as science. And then all research activities involving humans and other research with ethical implications. And they include genetic material access, undergo research ethics review. And this is something that is given by the international instruments. Nagoya says that. It says any material, any genetic material access research must go through ethics review. Are you going to collect those materials in an ethical manner? You know, ethics talks of three things: justice, benefit, and respect, isn't it? So are you respectful? Are you just? And is there a benefit? Are you respecting the fact that when you go to our pastoralist, that the knowledge of fermenting of the milk belongs to them and not to you? You might isolate the agent, but the knowledge of how they ferment their milk belongs to who? To them, isn't it? They may not tell you it is called bacillus, whatever you will come up with, but that knowledge belongs. Respect that. Be just to the fact that it is you went and found this thing happens in that community. And when you get benefit and patent, let that benefit also go back to that community. That is ethics. So you understand why every project must go through that is accessing genetic material. Because if anything comes out, the three elements of ethics need to be in play as you do your work. Then this is actually done to safeguard dignity, right, and safety and well-being of all participants. And that is the key thing of ethics. And that's why in ethics we say you cannot buy people. You can't go, like right now in Kenya, we have a bit of famine and drought. So you can't go to a community just because they are going through that and say, I'll be giving you each a sack of maize and this amount of money, come and. So people are giving their blood, not because they feel like giving their blood. Are you seeing the ethical issues? So you have to be sure that those issues have been completely dealt with, and yet you must also compensate the time that a person comes to the clinic or comes to where you are collecting their blood. They are not doing their work. They will use their transport to come. So that is where the ethics committee looks at and says, yeah, this amount of money is fair. It's just. You are not bribing, you are giving it. It's not a bribe to these people, but it is money that looks like fair compensation for the time you are taking to do the work. I've seen one of our ethics reviews, somebody had, I think, around a 4-page or 5-page questionnaire. And the ethics committee looked at the questions and they felt actually it will be taking around 2.5 hours because they were real detailed. You know, many questions with sub-questions. And they asked the PI, can you revise your questions? How do you expect somebody to come and sit with you for this long? Maybe do it in 2 phases. Do phase 1 and then go back to phase 2 so that you respect people's time. You respect people's privacy. There are some questions that are very private to people. So when you're asking that question, they'll tell you where will you ask the question. You cannot just sit in the open where other people are listening and you're asked, do you know, even asking somebody what is your age, they call it a very sensitive ethics question. How much money do you make from your milk? Sensitive ethics. You're talking about economics. Sensitive, personal to people. So that is when it's so important that the ethics reviews that and guides you. So if you say, I'll just be sitting at the market in the open, they tell you, no, you may need a room where these people come and you issue the questionnaire so that their privacy is maintained. So that is what is mainly. At ILRI, we have our ethics committee because we are headquarters here in Kenya and Ethiopia, registered in Kenya, registered in Ethiopia. And we are also registered in the US. Reason is donor funding. US needed us to register our ethics in the US. And it works for us. The EU accepts it. Australia accepts it. Most of the countries that give us donor funding accept it as an international registration. So that is why we get it accredited in those countries. So that is just membership of our ethics committee. And one of the important things is just to see that we'll always have an external member. At ILRI, our external member is mainly, and here we have quite a number of external members, people who are not part of ILRI, but they are members. This is a medical doctor, and this is a member of the community, elected by the community. We went back to the community, we wrote to them through administration, we told them we have an ethics committee, it does this, give us a member of your community who you think will represent you, and they send us a member. But it cuts across all the areas. So you can see it's heavy on animal and human health, but we have a legal person, because ethics, you must have a legal person. Then we have people from we have policy. These are people who do economics. We have sustainable livestock. We have animal and human health policies. So we have across our institution. And then we have research methods. Because you also look at how have people, how do you call it? The models of your experiments, isn't it? So somebody must look at it and say, are you using too many people? Or are you doing an experiment whereby even the numbers will not add up for you to say you have any information that you can publish? So that's why the research methods group is part of it. Then we go to the Institutional Animal Care and Use Committee. And this is mainly if you work with animals. And as you can see, a lot of you're working with animals. Mosquitoes are animals, isn't it? Immediately you touch a mosquito in health, you have touched an animal. And it puts you in the animal world. I can see Prof laughing. Well, that's an animal. So you need an ICO because you work with animals, you work with ticks, all these vectors are animals. They are not humans, they are animals. You cross over. Rodents are spreading this disease, so it means it's animals. You work models also in rodents and all that. Those are animals. So immediately you start working with animals, you need an ICOOC. And the ICOOC will mainly just review animal ethics and welfare. That is what they usually will look at. And it is just to ensure that there is humane treatment of animals. In ILRI, we make a joke and we say these animals, their cost per day is higher than all of us. Because if you just see what goes into those animals in research per day, and you look at how much you spend per day as the researcher, the cost of the animal is higher than your own personal cost. But it's supposed to ensure humane treatment of animals. And there are backgrounds to this. You can go back and read. Where did it come from? It came from a lot of suffering of animals in experiments in science. Even the ethics, if you look at the background of ethics, is people were doing experiments with people in prisons, and people suffered as experimental tools. So that is why all these things have come up, to just ensure that we do good science. We don't do science that is harmful to anyone. And our ICOOC is also registered in Kenya and is also registered in the US. And those are just our ICOOC committee member. We also, on ICOOC, we also have an external member on this committee, and then we have the secretariat, which is the UHS office. And then we have what we call the Institutional Biosafety and Biosecurity Committee, and we look at all activities involving use of hazardous materials. And at ILRI, we just define them as hazardous material. They could be biological, they could be chemical, they could be radioisotopes, they could be genetically modified materials, etc. And all this must undergo a risk assessment. So you must identify the hazard. I heard somebody articulate this morning that the gene drive hazard is what? If you are listening this morning, what was the hazard? What did he say that you must find out where it is? The construct. What is the construct? What is in the construct? What new are you adding? What are you introducing? That is what you must look at keenly and identify the risk. That is the likelihood and the consequence. What is the likelihood of that construct causing what he said it could cause? And what is the likelihood of that happening? So always remember, it all drives down to hazard and risk, consequence and likelihood. So you must always use a risk assessment as a tool to identify the risk, whether it is for recombinant DNA or any other biologicals. And it's important for us to understand that even when we are doing the risk assessment for recombinant DNA, what are you looking at? And maybe because none of you here is plant, so let me note. Is there a plant scientist? I don't think so. So a plant science example may not work much. When you are looking at, you are using it like in vaccines, in the animal work. I know the human work on vaccine, you are not regulated under Nagoya, so you might do a lot without having to go to NBA. But if you are in the animal world, or you are using your mosquitoes and now you have crossed over a little bit, what are you really looking at? You are moving maybe a gene from one animal to another, isn't it? Let me use an example of one that is happening at ILRI. I think as part of the research, the scientists have discovered, literature review, that warthogs are very related to pigs. True or false? Warthogs and pigs, they're actually almost the same. But warthogs don't get this disease called ASF. Pigs, that is what is killing our pig population in Africa and part of Asia. And it's even growing to a level that it might cross over to the other continents. So what is the work? They're trying to find out what is in a warthog that stops the warthog from getting African swine disease. And what do they identify? The genes in the warthog that make the warthog resistant to ASF. So then what do you do next? You pick those genes and see if you can put them in a pig and make the pig resistant to ASF. Isn't that not the genetic work you people are doing every day in your facilities? You are just trying, this is the gene, can I move it? The other one that they've tried, they've not gone very far, is on that word I always forget, trypanum. Yeah, and this morning it won't come. They realize baboons don't get that disease. When they go to the wild, they realize baboons never get sick with that disease. So what they identified, what genes are in a baboon that make them not get the disease. So if you pick that gene in the baboon and put it in the livestock, in the cattle, then the cattle will not get the disease. It will develop natural resistance. So that is what goes on, isn't it? But now you are in the GM work. all I'm saying, actually, those applications, Mushiri can confirm, they're all at NBA, isn't it? At least those two. We have applied to do that at NBA. So that is what you apply. And when you are doing the risk assessment, then you find out, when I pick a gene from the warthog, what else could this gene of the warthog be transporting to the pig apart from resistant to the disease? Is it bringing any other trait that could be of hazardous, especially to the downstream, to the food chain? Because the pigs eventually will be eaten by people. So that is what you're looking at. And then you do that full risk assessment. And we said, at the level of just finding out, can I actually pick a gene in the warthog and put it in a pig successfully? That one we are saying, that is discovery. It may not be highly regulated, but you must make sure that you do it in a lot of confinement so that there is no gene flow. With animals, it's easier. With plants, it's more hard. Now, once you have successfully moved the gene, then now you go to the next level, the confined field trial. How does this pig now behave after it has received this gene? Is there anything different? Does its protein, does anything in it change that could affect what feeds on the pig? So those are the risk assessments you do. So you identify the hazard, the gene, what is the gene doing, what changes now have you seen in the pig that has come up after those resistant, and then you come up with a good mitigation, write it up, submit it, proven with science, and you're good to go. So that is what the biosafety, that's what we talk about, risk assessment. And if it's just a normal pathogen, then you'll ask yourself, I'm working with Salmonella. What is the pathogenicity of Salmonella? Virulence of Salmonella. Once you can answer those two questions, you've done your risk assessment in simple terms, isn't it? And what will I be doing with Salmonella? Am I going to make pure cultures? What is the volume? How can I mitigate the Salmonella? You know, you can pick Salmonella, denature it, and drop it to open bench, because it is no longer harmful to anybody. So those are the things you do in risk assessment. That is why I'm never for this approach of it is risk group this, so you do in containment level 2. I don't think it is always a risk assessment. And with the labs that we have in Africa, we must do risk, because we may not have those high containment facilities, but we can innovatively work with these agents at lower containment. Maybe we've denatured, maybe we are working at low, or we are saying this thing is common in our environment, we already have it, it's not that we're introducing a new disease. There are many ways we can do our mitigation and drop the containment level. And this is mainly done to ensure that you secure safety of people and communities and the environment. And this is what is carried out by what we call our Biosafety Biosecurity Committee. So our approach is that holistic. We don't pick recombinant and have an IBC for recombinant and another IBC. No, our IBC looks at all these things. And as you've talked here, even the recombinant you're working on is malaria. So you're still thinking of malaria, Plasmodium, isn't it? How do you ensure that when I'm working with it, a mosquito will not be there, pick it and infect me as the worker working there, or it goes to the environment? I remember somebody saying it is easier to work with a mosquito than to work with a human being. So those are the things that you are really looking at IBC. What are the risks and which one can you accept, which one do you reject? So that is the membership of our IBC, and it has quite a number of external members. It is also constituted so that it can do recombinant DNA and any other risk. You know, it's quite interesting. The people we call experts of our IBC is just because they have extra knowledge in molecular biology to work with recombinant DNA. But when you look at it, they're either virologists, am I right? Or they are either molecular biologists. It's only that they have now this extra knowledge with recombinant DNA. So the same person who did the recombinant DNA risk assessment can do the risk assessment of working with Rift Valley fever as a virologist. True or false? So it's not that you change even the people in most cases, but sometimes you may need an expert if you don't have an expert in your committee. Then let's look at the area of permits and licenses. And these ones are determined by their country to country. Not all countries ask for the same things. But in most cases, what I've seen around is they include permission to carry research, permission to move biological materials. So to carry research is mainly science and technology or higher, Ministry of Higher Education. Then permission to move biological materials. If you are in health, it's public health. I hope I'm right. It's public health that gives you permits to move your materials, most of the time. And there's a department there that just pathology, I think. They are the ones who actually will give you a permit. If you are moving animal materials, then you go to the directorates of veterinary service in your country. If you are moving plant, you go to plant inspectorate service in your country. If you are moving wildlife, you go to the wildlife people in your country. You know, it's very interesting that we have plant, but if you're moving a tree material, you must get permission, especially in Kenya, from the forest department, because trees are regulated by the forest department. So you really need to find out who needs to give you what. And then there will also be permission to access genetic resources and indigenous knowledge. And this, in many of our countries, is under environment. It's only Uganda that I've seen them put all this under National Council of Science and Technology, and by the way, this makes it very easy to process permits in Uganda. They have one stop. You go to National Council of Science and Technology in Uganda, you'll get your research permit, you can move all your materials, they'll just refer you maybe to MIF for movement of material, but most of the permits they can give you at the National Council. But in most of our countries, this particular activity is under environment. So here in Kenya, you'll go to the National Environment Management Authority to get that permit. Then there is permit to handle animal, domestic, and wild. So if you're working with domestic animals, in many countries, some countries have not implemented this fully, but the countries that have implemented it, you need permission to handle an animal. Here in Kenya, you even need to be registered as an animal handler or a vet. So you must make sure on your experiment these people are part of it. And if you're working with wildlife, then you also must have registration to handle and work with wildlife. By the way, for wildlife, like in Kenya, you must involve the wildlife people. You must involve Kenya Wildlife Service. They do not allow anybody to work with wildlife. I've seen some of our PI come from abroad, talk like we've been working with wildlife. No way. They don't allow you to handle wildlife. You can go with them, but a Kenya wildlife person must be with you. And I think it's the same thing. I saw that on one project, somebody wanted to work with gorilla in Uganda, and it was the same thing. Their wildlife people had to be part of that project. Then there is, you need permission to conduct research using GM techniques. And I like just calling them GM techniques. I think they are techniques that we use, isn't it? Yeah, sometimes we say GM, but it's just techniques that we use. It is very rare. Very few of us are really going to modify a whole animal. Even the mosquito, you are not modifying. They call them GM mosquito, but have you modified the whole mosquito? It's just a technique you've used either to delete or insert or whatever you've done with that mosquito. And then permission to work with hazardous material. If you work with some hazardous material, even chemicals here in Kenya, you need, they call it poisons, and what board? In Uganda, you call it like the way the American call it foods and poisons board. You have something board in Uganda. The same name. Tanzania, what do you call it? If you're working with a very hazardous chemical, who gives you permission? The same people who register your drugs, they are called what? Like if you want to register, TMDA means? Yes, it's the same person who will register you if you're working. And if you go to their regulation, they have listed. I'm telling you, some of us never read it. Our regulators are not following them, but they are there in the regulations. They regulate and they tell you these particular chemicals are regulated in this country. So to work with that chemical, you need special permission to work with it. And it's there in most of our regulations. So those are the things. I'm just trying to navigate for you down, for you to understand how complex it actually can be. So you are in almost 7 ministries, different government departments, different ministries. And as I told you, they don't talk to one another. They really don't talk. So it's you yourself to have a way in which you can navigate them. So at ILRI, what we do is the Research Compliance Office actually does that navigation. And that's why, as I told you yesterday, our research compliance begins at your project proposal. So when you submit a proposal, even before it goes out for funding, it goes through the internal review, we map up the compliance so that we can see how the MARS looks like, how many countries do you want to work with. And I can tell you it is easy to work in a country where there is a compliance system than work in a country where there is no compliance system. Because if there is no compliance system, it means tomorrow a degree can just come up and they say we'll not work with it. Are you getting it? But when there is a compliance process, then you have a guidance document. Then I wanted to cover a little bit on Convention of Biological Diversity, the CBD, so that we understand where do some of these things we've been talking about so much really come from. They all come from the CBD, what is the Convention of Biological Diversity. And the CBD covers 2 protocols. That is the Cartagena Protocol and Nagoya Protocol. Cartagena Protocol gives you guidance on conservation of biological diversity resulting from use of modern biotechnology, and usually referred to as GMO. So the GMO work is what the Cartagena Protocol guides. And I always tell scientists, if you find yourself in a situation where there is no guidance, refer back to Cartagena Protocol if your country has signed it. Because most of our laws said we will comply with treaties that our countries are signatory to. So if you go back there, then you at least have something that can cover you. But that is where the GMO laws come from. Then we have the second protocol, which is the Nagoya Protocol, that gives guidance on access to genetic resources, fair and equitable sharing of the benefits. So this is what creates what we call access and benefit sharing guidelines that we see in a lot of the countries. So how do we comply to Nagoya? You ensure that you have all regulatory requirements for proposed project from the GM regulatory agency. And because you had even Mushiri saying that they keep changing. There are some things they stop. Yesterday I heard that, actually, Mushiri, is it true that now it's 6 months to submit our progress reports? I had it yesterday, 6 months. You know, I don't even know that I'm struggling to do my 3-month report, so maybe I should rest until 6 months are done. So they are really, they are not static. They keep on improving, or new regulations come in and they adopt them. So you always have to find out what are the regulations and what are you working with. Then confirm your facilities. Confirm that you have facilities that can implement. So that is where you start. What are the regulations in that country? If it's in Kenya, you go to NBA, find out the regulations. What do they require? Once you know the regulations, you come back to your own facility. And you confirm your facility can implement the project as prescribed by the regulatory authority. For example, in ILRI, our labs can do contained use. If you want to do a field trial, for animals, we have a big farm. We can do an animal trial. But we also do plants. You know, animals feed on plants. So if you work with animals, you must also work with plants. So when we want to do plants, then we have to get into a collaborative agreement with mainly our National Agricultural Research Institute, because they're the ones who own the big research farms in this country. Because that is where, you know, these people will tell you the distance of segregation is this much. Where will you get that distance in your own institution? So you must come up with where are you going to do your work. So maybe the labs, you have them. Sometimes you don't even have the labs. You may need to upgrade them. So that is why I'm saying you go back and find out, do you have the facility? Once you are happy with your facility, you call the regulator to come and review. You know the mistake most of us are making? We do everything, we even sent our document for approval, all the science, the risk assessment has been approved. Then the regulator says, before we start, let's come and see your facility. And they come and they tell you to do things that you have no money to do, to improve. So it's important that you do it early. You call them early to review so that you can negotiate and come up with innovative ways of doing that work. Then once you've done that, then complete your institutional research compliance process. Actually, at ILRI, what we do is we have a more detailed risk assessment form, more detailed than what the regulators give us. So in that, we give even guidance to our staff, and maybe later I will just share a copy of that, of one of them. So you will explain, you know, not everybody understands the risks of a construct. Why are we asking you? What should you show as you give this? What is the reviewer going to look for so that you give that information? So once you've done the internal process, you confirm review at compliance committee is in line with the requirements of country of implementation. So that is what IBC does. They make sure when you submit your form, they look at your form, we look at what has been done, and in the development of our form, we have made sure that it complies with the country where implementation will happen. Then we make application in the prescribed GM regulatory agency of the country of implementation. And the GM form in Kenya is not the same as the one in Uganda. Are you seeing the challenge here? So you have to go and pick the way the regulator, you cannot go and tell a regulator that my form is even more detailed. They don't take it. They're sitting here, they don't. You have to go and pick their form, whether you think it's inferior, you fill it the way they have asked. Actually, the first one we filled for animals was more or less a plant form. Can you imagine being asked to do gene flow in animals? Animals that you put in a bomber and you separate male from female. You even just mute them and nothing will happen among them. But you are being told to do gene flow like a plant. So you do it slowly, you negotiate with your regulator. But remember, you will have to put it in the form prescribed by the regulator, because that is what is in their regulations. So once you filled their form, you submit it to them. And when you are submitting, I think this is where some of us have issues. And I remember we had an issue in Tanzania with one of our applications for research. We applied, we sent it online, and we assumed they received it. You know that assumption, you get a receipt on your computer. But we never followed. So I wondered, why aren't they not responding? So to call them, they tell me, we've never seen that application. Maybe it went in junk. I don't know what happened to it. So I always say, if you send in your application, just follow up. Find out, have you received my application? It makes good sense. It removes a lot of headache. Confirm that your application has been received. And then wait for response from regulatory agency. And this is where I say, I don't take it from a PI to tell me I have talked to so-and-so. Government responds in writing. Am I right, those of you who work for government? Government never does gentlemen talk with people. For you to know you have a response from a government agency, it will come in writing. Nowadays, at least with COVID, they have agreed that they can send us emails, but they will write back to you. So ensure, even if they call you and tell you, Effie, it is approved, follow up until you get the written approval. That is the only evidence you have that your project is approved. Then note and comply with all approval conditions and implement your project. So those are the few steps that you just follow. Put those steps in a document, whether it's a guidance document, an SOP, and your scientists will have it. In ILRI, we have it in a flowchart. And that makes it easy for the people to follow. So when it comes to the CBD, the Nagoya has two. I told you it's Cartagena and Nagoya. So what does it? You complete your institutional research compliance process, and then now you go, ordinarily the process at country level includes you obtain a research permit, because they cannot give you an access permit without a research permit. And then you make a formal application for an access permit with the competent authority. And then the application for a negotiating. So in Nagoya, where you go for Nagoya Protocol, it has where you negotiate prior informed consent and mutually agreed terms. Prior informed consent means you've gone to the country and you've told them, I will be picking mosquitoes from this forest or from this area in your country for the purpose of research. So you are asking them prior to you going to pick. Prior informed consent is not what you go to ask for when the mosquitoes are in your laboratory. So as you do the prior informed consent, then you have to do what we call the mutually agreed terms. Those are the terms of collecting the genetic material. So they will tell you, now you are collecting mosquitoes from Kenya. Please note, these are Kenyan mosquitoes. You know, one day I was asked, and we were picking, you know the way we pick animals across the border? If you sample it in Uganda, it's Uganda. You go for pick and mat in Uganda. If you sample it in Kenya, it's Kenyan material. That is how Nagoya wrote it. So you can't say these animals cross the border every day. Wait for them in Kenya if you want them to become Kenyan material, sample them there. But if you are going to look for them in Uganda, they become Ugandan material. You look for them in Tanzania, they become Tanzanian material. So that is how it is written. So you entered, and this is done by the competent authority. In Kenya is the environmental authority. So they'll give you the prior informed consent. The environmental authority now in Kenya sends you back, if it is animal materials, they will send you back like to the Department of Veterinary Service. Because materials in the country have been given to specific regulators. So animals in Kenya belong to the government and they are held in trust with the veterinary department. So any animal, prior informed consent will start with the veterinary department. If you are going to the forest, forests are regulated by the forest department. If you are going to wildlife, Kenya Wildlife Service. So it changes that way. If it is marine, then you go to the marine. And that one is a bit complicated because even the borders are not very clear. So the negotiation sometimes can be very complicated with marine. But it's just agreeing that the terms of collection, you to note that if anything good comes from your research, you will give benefits back to that country. Those people will benefit from that research. And then you enter, after that, if you need to move any materials, you enter into a material transfer agreement with involved parties. Most people do this when they are moving material from one country to another. At ILRI, we realized it became so complicated. So in ILRI, we do material transfer agreement between ILRI and KEMRI, ILRI and KALRO, that is the Kenyan Agricultural Research. Anywhere, anybody we are sharing material with, or we are picking their material, we do a material transfer agreement because it defines what IP belongs to that institution. So that you don't start arguing at publication. What was my IP? What is yours? I gave you my material. I had done the following. You agree all those things in an MTA, and then you can move the material. Then there is also reporting that you need to do. You need to report any incidents or deviations from protocol to the Research Compliance Office. And in ILRI, it is the Research Compliance Office because we coordinate it, and we will report this back to the Research Compliance Committee. So if it's animals, we'll take it to ICOOC. If it's livestock, if it's ethics or working with humans, an incident, it will go to the Ethics Committee. If it's a lab-acquired incident, we'll take it to IBC. Then you also need to submit a yearly or end-of-project report to our Research Compliance Committee through the Secretariat. So we ask every project to give us a yearly report. And the reason is the regulators are asking us for such reports, isn't it? So we ask, and we know that some projects go for 3 months, for 6 months, so either yearly or at the end of your project. And then we submit a progress report to regulatory agencies as prescribed in approval letter. All the approvals letters, even the ones for research permit, it tells you, give me a yearly report. The one for NBA used to tell us to give 3 months, I'm told it's now 6 months. So I'm sure in our next approval, they'll tell us, give us reports every 6 months. And then the final one is the monitoring and evaluation. And this involves internal monitoring of projects for compliance, to institutional, donor, and country approval conditions. So we do our own internal monitoring. It is a legal requirement, and every year accredited compliance committees must submit an M&E report to the government authorities. So National Council of Science and Technology, if you have a registered ethics committee, expects you to give them a yearly report of M&E. And the good thing is they don't expect you to review each and every project. They ask you to just sample 1 or 2. Audit them, give them a yearly report. When it comes to NBA, they don't ask us actually to do M&E, but if they are coming every year to do your M&E, you are better off checking yourself, true or false. Do you want a regulator to come and check you when you have never checked your own system? I don't think it works well, so it's just important also to do it. And then we have M&E from the GM regulatory agencies at country level. So those are the monitorings that we usually would have. I'm sure you can't read that, but that just shows you our compliance form, and that's why I didn't want to share them. I realized nobody will read them on the screen. But it's just a simple way of helping a scientist feel this from a risk assessment point of view. It's asking simple questions, you answer them, we are able to get the information we want. So when it goes to ethics, it's an ethics application form. I think I've seen the ethics application form for Cambridge. It's risk-based when I look at it, because they ask you questions, you respond to the questions, and then it goes to the committee. Now with those questions, we develop what we call, at ILRI we have what we call review tools, because we want our reviewers to use the same tool. So we have review tools. So the review tool will give question 1, what should you as a reviewer make sure you have looked at? You can add many more things, but make sure you give us comments on the following. So that way you fill your comments, and those are the comments we put together as the review comments. And then we'll send them back to the plenary for full agreement and make a decision for approval for your research. And I think that's the end of it. So I hope you have had a little bit of a more broader, more understanding of how the research compliance process in science works. And it can take a lot of different anchors. I've talked a lot on livestock, but because of One Health, we've had ourselves also work with a lot of the public health department, because we have to apply for our research with public health with them, with human subjects with them. And they have their own process, and you have to go by it. And I think, finally, what I want just to talk about, I wanted to ask you a question at the end of this. If you are working with those recombinant mosquitoes, and that is human health research, do you really need NBA? Do you need to go to the regulatory agency for GMO? It's malaria. Malaria is a what disease? Human disease. You are doing it for the purpose of human. So do you need regulation? Nagoya, yeah. You know, the CBD put you off, said human research is not covered by this. By the way, the reason why human research was not covered by CBD is because you are covered under GCP. You do detailed GCP in your good clinical practices in your research. So do you need to go?
Yes.
Why? Give me a reason why you need to go.
Well, because I'm here.
Put on your microphone so that they can translate for those who are French speaking. Or where are you? I give you my own. Okay.
Okay. Yes, we still need to go to NBA because even if the target disease is malaria, the target organism is mosquito, and its movements and whatever you want to do with it is also regulated by a different body. In our case, in Mali, maybe it's maybe different in different countries. So it's the Minister of Environment. So you need to go to that body, government body, to get the authorization, the needed authorization. So NBA, in our case, is under the Minister of Environment. So yes.
You need that. Anybody else who wants to add on that? Yeah?
Yes, I'm speaking as a regulator. And this is how I see it. they are not doing human research. They are doing animal research to solve a problem in human beings. Basically, the research has to be with animals. And they need to go to the regulator, specifically the NBA, because it is a genetically modified research. And so that is why they need to go to the NBA. And when they succeed there, then it is applied at the human level. The problem with the humans, they know already, and it is malaria. But the how is what is happening at the animal level. So that is how I see it as a regulator.
So I think for the health people, yet when you are doing RVF vaccine development using recombinant techniques, you don't apply to anybody. Am I right about that, Kemri? Do you apply to anybody? Vaccine development, diagnostic development, using recombinant techniques. They don't, because it's mainly focused. They're just picking genes, transferring all the trials, everything is in a human being. These are the areas that I call gray. So because they might be working with RVF, Rift Valley fever, the virus, you are doing recombinant techniques, you are trying it in human beings as a vaccine, but that doesn't fall under CBD. But when you pick a whole mosquito and modify it as a mosquito, it falls under CBD. Because now you are working on, as he says, you are picking an animal and modifying the animal to help you solve a human disease. So it's always important, and that's why IBCs are so critical. Because even you in human research, you need to take your project to IBC, and IBC should be able to navigate and tell you, no, this one we think we need to ask the GM regulator, whether it falls under GM work or not. And you'll always come with those kind of issues, isn't it? When you work with ticks, if you modify the tick, I don't know, it may need to go to GM for GM regulation if you modify the full tick. But if you just pick the salivary gland and modify this, so those are the issues that you have to discuss. What I'm trying to tell you, the regulation on GMO, especially when it crosses over to human research, and you're using, because GM also, you regulate microorganisms. In Kenya, you do mushiri, isn't it? And you people work with microorganisms, recombinant DNA, but really they are not regulated when you look at it that way. So those are the gray areas that even as regulators, you need to make them more clear. Because when I read the regulation, it says you exempt human research. Very clear in the law and in the regulations. So we need to be a little bit clear on which area, and the IBCs need to be even thoroughly trained so that they are able to tell which one is exempt and which one is not. Thank you.